How did life emerge from an inanimate world? This question has intrigued humanity for centuries. The mystery of life's origin is not just about understanding our past, but also about deciphering the fundamental laws of nature. Our journey will take us back billions of years, from the early Earth's fiery birth to the emergence of the first life forms. We'll explore the building blocks of life, the experiments that brought us closer to the answers, and the theories that guide our understanding today. So ready to step back billions of years and traverse the path from lifelessness to life? Imagine a world far removed from the green and blue planet we call home, a primordial land of volcanoes, thunderstorms, and vast oceans. This was Earth over 4 billion years ago, a young, fiery planet shrouded in a thick atmosphere composed primarily of nitrogen, carbon dioxide, and water vapor, with barely any oxygen to speak of. The Earth's surface was a chaotic theater of geological activity. Volcanoes erupted, spewing ash and molten rock into the air. Thunderstorms raged, lighting up the sky with their electric fury. And beneath it all, the Earth's crust was constantly shifting and reshaping itself. The oceans of this early Earth were far from the tranquil blue waters we know today. Instead, they were hot, acidic, and filled with dissolved minerals. These oceans were more like a global chemical soup, stirred by the planet's relentless geological activity. Yet, it was in these extreme conditions that the magic happened. The raw materials for life, such as carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, were all present. And the energy from sunlight, heat from the Earth's interior, and the electric charge from thunderstorms provided the spark needed to drive the chemical reactions that produced the first building blocks of life. In this seemingly inhospitable world, the stage was set for a remarkable event: the birth of life. Life as we know it is built from a few simple molecules: amino acids, nucleotides, and sugars. Let's take a closer look at these building blocks. Amino acids are the architect's bricks, the very foundation of proteins, which are the workhorses of every living cell. They carry out a myriad of functions, from catalyzing metabolic reactions to replicating DNA. Nucleotides, on the other hand, are the scribes. They form the core of our genetic material, DNA and RNA, carrying the blueprint of life from generation to generation. And finally, sugars, the energy merchants, providing the fuel for all of life's processes. Now you might wonder, how did these molecules form on the early Earth, devoid of life as we know it today? The answer lies in the primordial soup theory, a popular hypothesis suggesting that these building blocks were synthesized from simple inorganic compounds like water, ammonia, methane, and hydrogen present in the early Earth's atmosphere. Under the influence of energy sources like lightning and solar radiation, these molecules could have engaged in a series of chemical reactions, leading to the formation of organic compounds, including amino acids, nucleotides, and sugars. These molecules, formed by natural processes, were the first step towards the complexity of life. They set the stage for the next chapter in our story, the Miller-Urey Experiment, where scientists recreated these conditions in a lab, giving us a glimpse into the dawn of life. In 1952, a groundbreaking experiment brought the primordial soup to the laboratory. Stanley Miller, a young chemist under the supervision of Harold Urey, set out to simulate the conditions of the early Earth in a laboratory setting. Their aim was to test the hypothesis that the simple compounds present on early Earth could have combined to form the complex organic molecules necessary for life. Miller constructed an apparatus that included a flask of water, representing the ancient oceans, and another flask filled with a mixture of gases, thought to represent the early Earth's atmosphere. These gases included methane, ammonia, and hydrogen. A continuous spark, simulating lightning, was introduced into the system. Over the course of a week, Miller observed that the clear solution in the water flask had turned a murky brown. This brown residue contained amino acids, the building blocks of proteins, and a fundamental component of life. The Miller-Urey experiment was a game-changer, a testament to the power of scientific exploration. It demonstrated that given the right conditions, the building blocks of life could form spontaneously from simple compounds. This pioneering experiment has been replicated and expanded upon, with similar experiments producing nucleotides, the building blocks of DNA and RNA, and even complex carbohydrates. The Miller-Urey experiment proved that the building blocks of life could indeed arise from the conditions of early Earth. It was a significant leap forward in our understanding of the origins of life, and it continues to inspire scientific exploration into this fascinating question. The first self-replicating systems might not have been DNA-based, but rather RNA-based. This intriguing concept forms the basis of the RNA World Hypothesis, a tantalizing model for the origin of life. In the grand tapestry of life, RNA is often seen as the middle child, bridging the gap between DNA and proteins. But what if in the early stages of life's existence, RNA was the star of the show? The RNA World Hypothesis posits just that. This hypothesis suggests a world where RNA molecules, not DNA, were the first self-replicating entities. RNA, with its ability to both store information like DNA and catalyze chemical reactions like proteins, could have served as both the blueprints and the builders of early life. Now, you might be wondering, how could RNA have catalyzed its own synthesis? It all comes down to the unique capabilities of RNA. Some RNA molecules, known as ribozymes, can act as enzymes, speeding up chemical reactions. In the RNA world, ribozymes could have catalyzed the formation of new RNA molecules from nucleotides in the environment. This process, however, isn't without its challenges. For one, RNA replication is an error-prone process, leading to mutations. While some of these mutations could have been beneficial, sparking evolutionary changes, others could have been detrimental, hindering replication. Another challenge is the origin of RNA itself. How did the first RNA molecules form from a soup of simple chemicals? While we don't have all the answers yet, research suggests that the building blocks of RNA nucleotides could have formed under the conditions of early Earth. Despite these challenges, the RNA World Hypothesis has found support in various quarters. For instance, the discovery of ribozymes and their catalytic abilities bolstered the credibility of the hypothesis, shedding light on how RNA could have played a dual role in the early stages of life. In conclusion, the RNA World Hypothesis offers a compelling model for the emergence of self-replicating systems. It paints a picture of a world where RNA, not DNA, took center stage, catalyzing the dawn of life from non-life. It's a reminder that life, in all its complexity, may have sprung from humble beginnings. The transition from an RNA world to a DNA protein world marked a pivotal moment in life's history. Imagine, if you will, a world of RNA molecules self-replicating and catalyzing chemical reactions. Over time, these RNA molecules evolved, growing more complex and diverse. But there were limitations to what RNA could achieve on its own. Enter DNA and proteins. DNA, with its double helix structure, offered a more stable and reliable way to store genetic information. It was less prone to errors during replication and could carry more information given its larger size. This was like upgrading from a basic filing system to a state-of-the-art database. And then there were proteins. Proteins are the workhorses of the cell. They perform a staggering array of tasks, from speeding up chemical reactions as enzymes, to serving as structural elements, to transporting molecules across cell membranes and much more. Proteins added an entirely new level of functional complexity. But how did this transition occur? One theory suggests that RNA molecules, which can act as both genes and enzymes, started to synthesize proteins. Over time, these proteins took over many of the roles previously performed by RNA. Meanwhile, DNA began to take over the role of storing genetic information. This transition paved the way for the diverse life forms we see today. From microscopic bacteria to towering trees and complex animals, the emergence of DNA and proteins was a turning point, setting the stage for the incredible biodiversity we see in the world around us. From self-replicating molecules, the next step was the rise of the first cells. But how did these complex structures come to be from simple molecules? That's a question that has puzzled scientists for centuries. Today, we delve into some theories that might hold the answers. Let's start with lipid bilayers. Lipids are fat-like molecules that have a unique characteristic. One end of a lipid molecule is attracted to water, while the other end is repelled by it. When placed in water, lipids naturally arrange themselves into a double-layered structure, with the water-loving ends pointing outwards and the water-hating ends tucked safely inside. This structure, known as a lipid bilayer, could form the basis of a cell membrane, creating a tiny bubble within which life's chemistry could take place. But that's only the beginning. For life to thrive, these lipid bubbles needed to become more complex, developing ways to control what enters and leaves them, and creating an internal environment where chemical reactions could occur. This is where hydrothermal vents might come into the picture. These underwater chimneys spew out hot, mineral-rich water, creating a gradient of temperature and chemicals that could drive the formation of more complex structures within the lipid bubbles. Imagine, if you will, a world of hot, bubbling vents on the ocean floor, surrounded by a soup of organic molecules. Lipid bubbles form, grow, and split, gradually becoming more complex as they interact with the chemicals around them. Over time, some of these bubbles might have developed the ability to replicate themselves, essentially becoming the first primitive cells. This is, of course, just a theory. The real story of how the first cells emerged remains a mystery, but it's a mystery that scientists are working tirelessly to solve. As we continue to explore the origins of life, we come ever closer to understanding our own place in the grand tapestry of existence. The emergence of the first cells was a crucial milestone in life's evolution. As we move forward, we'll see how these simple cells gave rise to the vast complexity of life we see around us today. But that's a story for another time. A profound shift in Earth's atmosphere, the Great Oxygenation Event, forever changed the course of life. This pivotal event began over 2 billion years ago when the Earth's atmosphere, once devoid of oxygen, started to fill with this life-giving gas. The architects of this atmospheric transformation were none other than early forms of life themselves, cyanobacteria. These minute organisms, harnessing the power of the sun, began to photosynthesize, a process that releases oxygen as a byproduct. This oxygen began to build up in the atmosphere, sparking a revolution that would forever alter the trajectory of life on Earth. Before this event, life on our planet was anaerobic, meaning it existed without the need for oxygen. But the new oxygen-rich environment was a game-changer. Oxygen, a highly reactive element, allowed for more complex biochemical reactions and thus more complex forms of life to evolve. Yet this increase in oxygen was not without its challenges. It posed a threat to the existing anaerobic life, leading to Earth's first mass extinction. However, this upheaval eventually gave way to an explosion of diversity, as life evolved to not just survive but thrive in this new oxygen-rich world. This is the oxygen revolution, a time when life itself reshaped the planet in profound ways, paving the path for the emergence of diverse and complex life forms. The oxygen revolution opened up new possibilities for life's diversification. From simple cells to complex organisms, life on Earth embarked on a journey of endless diversification. This journey was not merely a random sequence of events, but was guided by the principles of chemical evolution. This evolution, which began with the formation of the most basic building blocks, set the stage for the grand spectacle of biological evolution. Chemical evolution, in essence, is the process through which simple substances gradually developed into complex compounds capable of facilitating life. These complex compounds, such as nucleic acids and proteins, formed the foundation of the first cells. As these cells multiplied and adapted, they began to form increasingly complex structures, finally culminating in the diverse life forms we see today. But this transformation did not occur overnight. It was a slow, meticulous process spanning billions of years. Each step was crucial, building upon the last, setting the stage for the next. From the formation of the first amino acids, to the development of self-replicating RNA, to the emergence of DNA, each milestone marked a significant leap in the journey towards complex life. As the complexity of life increased, so did its ability to adapt and survive. This led to the diversification of species, each uniquely equipped to thrive in its environment. From the deepest ocean trenches to the highest mountain peaks, life found a way to flourish. Thus, the seeds of life planted billions of years ago blossomed into the astounding diversity we witness today. Understanding our origins is more than an intellectual pursuit. It's a journey to comprehend our place in the cosmos. This journey has led us from the volatile environment of early Earth through the complex dance of chemical evolution to the dawn of life as we know it. Each step, each discovery, paints a more detailed picture of our beginnings. Today, we stand on the shoulders of giants, building upon centuries of scientific inquiry, yet the quest is far from over. The mysteries of life's beginnings continue to captivate us, pushing the boundaries of our knowledge and technology. Our curiosity propels us forward as we search not only for answers here on Earth, but also beyond our planetary borders. As we unravel the mysteries of life's beginnings, we come closer to understanding not just where we came from, but also where we might be heading. Don't forget to like, share, and subscribe for more journeys into the wonders of our universe.